To the day 4 video. This is the last day we're going to spend on this lesson, and next week we're going to be starting a new lesson. Alright guys, so we spent the last few days talking about light microscopes. Remember that light microscopes can only reach a magnification of 1,000 times. After you get to 1,000 times magnification, things start to get blurry. This is why you have to start using the oil immersion and stuff like that. And even then, if you try to go above 1,000 time magnification, your images would be so blurry you would hardly see anything. So for this reason, scientists invented a new type of microscope called the electron microscope. These have actually been around for a long time, but they are amazing microscopes. They can allow you to see things up to even 1,000,000 times zoom. Some of them up to 500,000. The newer ones, you can zoom in about 1,000,000 times. They're really, really amazing. So basically the way that they work is instead of using light, they take electrons, they bounce them off the sample, and then a computer, once the electrons hit the sample, they bounce off, the computer catches them, and it recreates an image of what the actual sample would look like. So you're not directly looking at the sample, you're looking at a computer-made model of the sample. And the only thing is, unfortunately, even though electron microscopes are amazing when it comes to zooming in very, very, very deeply, like using electron microscopes, we can see organelles inside of a cell and even viruses and teeny tiny little things that no other microscope can actually help us see. Even though all of that is true, electron microscopes only give images in black and white, and most of them can only be used on dead cells. So you have to kill the cells, you have to stain them with heavy metals, which are pretty dangerous. You have to slice them very thin, and then finally you can actually look at the sample. But you can zoom in a lot deeper. So let's just get an idea of the size of things. Here we're at about ×2. This is ×10 magnification. Let's see what's on the pin. At around ×100, we can see a human hair, and we can see dust mites. Dust mites basically live everywhere. They're like teeny tiny little spiders. You don't have to be afraid of them, but some people are allergic to dust mites, so whatever. Anyways, so if we zoom in up to ×100, we start to get the first appearances of something down there. So let's see what these could be. Let's go deeper, 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 and now we reach ×1,000 zoom. So this is the best that a light microscope could ever possibly reach, right? ×1,000. After that point, images start to become blurry. So let's see what these could be. So we have ragweed pollen. This is basically just a piece of pollen that comes off of a plant. We have a white blood cell, which is found inside of our blood. It helps to kill bacteria and viruses and stuff like that. And then we have our red blood cell. And then we have yeast. All of these three things can basically be used using a light microscope. However, they will be blurry and you're not going to get a whole lot of detail on them because they are past the 1,000 times magnification. If I want to see something smaller than this and I want to get real detail, then I have to go further and I have to maybe switch over to an electron microscope instead of using a light microscope. So up until this point, the light microscope has done us well. We're totally fine. We can still use it. Now we have to switch over to a different type of microscope to get real detail. So let's go here to the ×10,000 magnification. Here I can see E. coli. E. coli is a type of bacteria. Guys, in case you're wondering, eukaryotic cells, which are like human cells, plant cells, and multicellular organisms, our cells are clearly way, way, way bigger than bacteria cells. Bacteria cells are tiny and you actually cannot get a lot of detail out of them using a light microscope. You might need to use an electron microscope to actually get real detail. So these two are the bacteria cells, right? So we have E. coli and we have staph, right? And then if I want to zoom in even deeper and see what this might be, when we get to ×100,000 times zoom, we get to the Ebola virus. So the largest things that we can see are like dust mites and stuff like that. And then we get to our eukaryotic cells, which are like plant cells and animal cells and yeast. And then we get to bacteria, which are even smaller. And then finally, we get to viruses. Viruses are absolutely tiny, and you cannot see them unless if you use an electron microscope. Look at this. We had to go in times 100,000 times just to see the Ebola virus. And the Ebola virus is actually considered a pretty big virus compared to most other viruses. So now let's see what's down here. Now we have zoomed in 1,000,000 times to get to the rhinovirus. If any of you guys are wondering, this is the virus that causes the common cold. But can you imagine how tiny this is, how difficult it was to actually discover what a virus is? Viruses are way smaller than bacteria, and the only way that we can actually see them is to use an electron microscope, right? So this is why it's so important to have different types of microscopes. All right, so back here, we now know why we use electron microscopes. Let's talk about how electron microscopes have changed over the years. So our first type of electron microscope was a transmission electron microscope. Using this, we were only able to get 2D images only in black and white, and the specimen had to be dead. So we could never see the actual organism moving and living and seeing how things actually work using this type of microscope. But they were amazing for us to actually be able to go into a cell and look at organelles and look at viruses and stuff like that. They still had an incredible ability to zoom in on specimens. However, there were a lot of disadvantages. And then a new type of microscope was created, the scanning electron microscope. The same situation, your specimen had to be dead, which means you could not work with live specimen. It also produced images with no color, but it allowed you to see images in 3D. So you can see that this is a cell that has been cross-sectioned off, just cut into a thin slice, and this is a 3D image of a red blood cell and a white blood cell. And again, it has no color. And finally, the newest evolution, according to your book, is the scanning electron microscope. Now this scanning tunneling microscope, sorry, this was a mistake. I will go and fix that. So the scanning tunneling microscope is the newest type of microscope that we're going to talk about. It allows you to create 3D images. However, it is not colored, but you can use it on live specimens, which means that we can use it to actually see living things and how their organelles move and how things interact and all of that. Now, this is actually such an amazing microscope because it doesn't just let you see like little organelles and viruses. It can let you zoom in down to an atomic level. So you can actually see atoms and molecules inside of an atom, inside of a cell, which is pretty amazing. It's time to start talking about actual cells. After all of this discovery using microscopes and using the electron microscopes and looking inside the cell with the electron microscopes and outside the cell with the light microscopes and all of that, several things were discovered about properties that all cells share, or that almost all cells share. So, all cells have a plasma membrane, meaning on the outside you have this plasma membrane, right? It keeps all of the stuff inside the cell. It prevents everything from floating out. Even plant cells have a cell wall, but right underneath that thick cell wall, there's a plasma membrane. So it's just like another wall that prevents things from moving in and out of the cell. Even this little bacteria cell right here also has a cell wall, right? Also has a plasma membrane which helps to hold everything inside of it, right? Another thing that all cells share is that they all have cytoplasm. Cytoplasm is the jelly inside the cell. So all of this blue stuff is your cytoplasm. So they're all filled with liquid and they all have a plasma membrane. The third thing is almost all cells contain genetic material. So here you see that there's genetic material inside the nucleus of plants and animal cells. And in bacteria cells, this right here is the genetic material. It's not inside of a nucleus. It's just kind of floating around all over the place. Okay? The only reason they say almost all cells is because there is a very small number of cells that do not have genetic material, and usually those cells are part of a multicellular organism, so they survive because other cells are giving them the things that they need to survive. But on their own, without genetic material, you cannot survive. Okay, this is why it's almost all. Almost all cells break down molecules to generate energy, which means all of these cells have machines that allow them to break down sugar or other molecules to make ATP or other forms of energy. And again, the only reason it says almost all is that if a cell does not have this ability, it's because it lives in a multicellular organisms where other cells are helping it get its energy. But if that cell that didn't have these machines was on its own, it would die immediately. So these are the properties that all cells share. So just to make it easier, ignore the word almost and just say all cells, but we need to know that there are some exceptions to these two rules. All right, so the first type of cell we're going to be talking about are eukaryotic cells. Eukaryotic cells are like plant and animal cells and yeast cells and stuff like that. They have a nucleus which helps to hold all of their DNA inside. The nucleus is a very important organelle. It helps to protect the DNA and control it, and it's basically the brain of the cell. So you'll see here a nucleus and another nucleus. Very important. Another thing about eukaryotic cells is that they have organelles. See all of these teeny tiny little things floating around? Those are like little organs. Each one of them has a specific job that it does inside of the cell. So they have organelles, right? Another thing, eukaryotic cells are usually much larger than prokaryotic cells, like what you guys saw in the simulation from before when we zoomed in on cells and then on viruses. First of all, we saw the eukaryotic cells, like the red blood cell, and then we saw the bacteria, and then we saw the virus. That's because eukaryotic cells are way larger than bacteria and viruses. Okay? And the last thing, eukaryotic cells are usually multicellular organisms, meaning like us. We are made up of many cells that live and work together, but they can be unicellular sometimes. So for example, yeast, the stuff that you use to bake bread, that is a unicellular eukaryotic cell. But in general, eukaryotic cells are multicellular. They just can be unicellular in some cases. The next type of cell we need to talk about are prokaryotic cells. Prokaryotic cells are thought to have been the original cells to exist on Earth. They are very simple. They're not extremely complicated. They're not like the eukaryotic cells. And like what you guys saw in the simulation, they are much smaller than eukaryotic cells. So the defining features of prokaryotic cells is that they don't have a nucleus and they don't have organelles. So if you look here, this is its DNA. It's not enclosed inside of a nucleus. It's just free to go wherever it wants throughout the cell. And it does not have membrane-bound organelles. See, these are organelles inside of a eukaryotic animal cell, but you don't see any of that in here. Instead of having organelles, they just have little machines that do their job in random places around the cell. Okay? So they are much smaller and simpler than eukaryotic cells, and most unicellular organisms are prokaryotic. So if you ever look at a unicellular organism, one that it's made up of only one cell, you know there's a very good chance that it's prokaryotic. It doesn't have a nucleus, it doesn't have organelles. It's most likely not going to be eukaryotic, right? So now, a lot of scientists wondered, how did this happen? How did all of these amazing things show up in the eukaryotic cells? What happened during evolutionary history for this kind of thing to happen? They realized that bacteria are around the same size as the organelles inside of a eukaryotic cell. So this is obviously way zoomed in, but this is around the same size as this guy right here. So scientists tried to think about what could have possibly happened. How did eukaryotic cells even exist? Prokaryotic cells are very simple, so they must have been the originals. Eukaryotic cells must have shown up later on. So scientists believe that there's a theory called endosymbiosis. As you guys remember, symbiotic relationships are relationships like mutualism where two organisms live together and benefit from each other, right? So let's say this is the ancestral prokaryote. This is a prokaryote that is living by itself. It has its own situation, it has its own DNA and everything. Then, all of a sudden, different parts of the membrane began to fold in and we started to get a nucleus. And then, this is where it gets very interesting. This is the theory of endosymbiosis, is that different types of bacteria started to come and move into this other prokaryote. So this is one prokaryote and these are other prokaryotes. They came into the prokaryote and as it turns out, the relationship was good. So this prokaryote allowed them to keep living inside of it, right? So let's say this is the mitochondria. He used to live by himself. He did not, he was just out there being a powerhouse, making energy. And this is a cytoplasm, a chloroplast. He was also out there doing photosynthesis, living on his own. When these two moved into this cell, they started to help this cell, and this cell started to help them. And for that reason, this cell had an advantage in life, which allowed it to continue to evolve and survive because suddenly it was getting all of its energy from these guys, and these guys were also benefiting from him. So this is the basic idea of a eukaryotic cell. It all started off as a bunch of prokaryotes swimming around independently, and then they all fused together and started living together forever because the relationship was so beneficial. All right, guys, so that's the theory of endosymbiosis. So, end of day 4. Remember, today is the quiz, and the quiz is going to cover parts of the entire lesson. So go over the video, go over previous work, make sure you get the basic idea before you actually start today's quiz. And good luck.